Okay, so I'm just going to stop the transcription because that's slightly annoying. There we go. So I'm Lindsay, I'm the Patient and Public Involvement Lead for the PHOSP-COVID study. Can we have the next slide, please, Tom? So we've got a jam-packed agenda for the next hour. We're going to have a series of presentations from the lead investigators in the PHOSP-COVID study, and also at the end we're going to have a panel discussion. So please do submit your questions using the chat box. We've also had some questions that have been submitted through Eventbrite, so that we can come to those as well. I'm just going to get the panel to introduce themselves. So I'll start with Chris Breitling.
Thanks, Lindsay, and I'd extend my welcome to everybody as well. And I'm Chris Breitling. I'm a respiratory physician, and together with Rachel and Louise, who introduce themselves in a moment, co-lead the study. But it is really the size and scope of the study that's been quite phenomenal, with how many investigators have been involved, from senior scientists, people working in the clinical side, people who are really trying to understand the mechanisms of long COVID as well as what to do next. And we'll be telling you a lot about that today. So thanks, Lindsay, for setting this up.
It's all right, thanks, Chris. Rachel Evans.
Hi there, everyone, and thanks so much for your time this afternoon to join us. So I'm a respiratory consultant at Glenfield Hospital, a clinical academic, and I also lead our long COVID service.
Thank you, Rachel. Louise.
Hi everyone, thanks for coming along today. I'm Louise Wayne, I'm a Professor of Respiratory Research at the University of Leicester. Unlike Chris and Rachel, I'm not a medical doctor, I'm a scientist, but I'm really excited to be here and to talk to you about the science today.
Super, and I'm hoping that Claire Hasty is here as well. Claire's our Patient and Public Involvement member. Are you here to introduce yourself, Claire?
Yes, I'm Claire Hasty. I'm a founding member of Long COVID Support. Our support group has 60,000 plus members, and we do a lot of research involvement work with policymakers and the media. Glad to be here. Thank you.
Super. And we've also got Ananga and Tom, who are kind of our behind the scenes tech support for today. I will just ask you to mute yourselves if you're not speaking, if that's okay. So just to sort of set the scene, we've got a patient video that was put together by Asthma and Lung UK, just about experiences of living with long COVID. And I hope that puts into context all of the work that we want to tell you about today. So Tom, if you just stop sharing and we'll share the video, thank you.
Before the pandemic, I lived independently. I lived in a flat share with two other friends. I worked as a British Sign Language interpreter. I was freelance, so I travelled here, there and everywhere for multiple bookings per day. And due to being freelance, I'd also take time off every year and go backpacking. Within my work and my career, I had a very busy, kind of 100% fulfilled life, really. I caught COVID in March 2020. I was 30. It started off with what I thought was a chest infection. I'm asthmatic. For 3 weeks, I had what I'd call active COVID, but for 7 weeks, I still couldn't walk. So I was struggling just to walk around the house due to muscle weakness and fatigue. It wasn't until that October that my doctor had coined the phrase long COVID, which is something I'd never heard of. So it was a very, very scary time. Having long COVID hasn't just changed my life, it's changed everybody's lives around me. A lot of my friends and my mum feel like they're grieving for my old self. My mum has had to change her career in order to care for me. It impacts everybody around you. My long COVID symptoms are continuous breathlessness. I also get heart palpitations. I've been diagnosed with POTS, so my blood pressure doesn't regulate itself. I get really severe fatigue. So if I do anything one day, I know the next 2-3 days have to be a rest day. I get really bad pain throughout my whole body and my joints and muscles. I get tremor in my hand that they can't explain, chronic migraines, dizziness, and it's my mobility really that's been impacted. So I use a walking stick or a wheelchair to help me be independent. I think more research is needed into long COVID because there's still so many unknowns. I think research would give it validation, but also to have some kind of treatment and some kind of cure would give people their lives back. I never thought at 33 I would be back home and my mum was my carer. It's so difficult to explain to somebody how much my life has changed, but just to even have 25% or 30% of my old self back would just be incredible.
Thanks for sharing the video, Ananga. If we just come back to the slides, I'd like to invite Chris to start, just to give us an update on where we are. I know obviously we had our last webinar was last August, so just an update on the progress really of PHOSP over the last 12 months. Thanks, Chris.
Thanks very much. I mean, very powerful video, and I'm sure many of you who've joined today, that may be quite a familiar story. So really what we want to do is really tell PHOSP-COVID story as to where we are now. And on to the next slide. So we really started the idea that we anticipated that, unfortunately, following the acute COVID episode, that there may well be some long-term sequelae. And that then led to the PHOSP-COVID study, which was focused on those who were hospitalized, in part because we anticipated they might be those who were most affected, but also because at that time they were the only ones who were being tested. So it was the only ones we could say with absolute certainty that they'd had a case of COVID. And then late summer, we then actually started the study in 2020. So we're now approaching 3 years on. We had a very broad vision, and the vision was to establish a national platform to integrate research and clinical service, because as you can understand, there was no clinical service because this condition didn't exist prior to COVID. And really to then try and understand and improve the long-term outcomes for survivors who are hospitalization with COVID-19. And the next slide. Even though, as you would have already heard, the leadership really came from respiratory specialists, but the reason for that was obviously the acute condition started very much as a respiratory infection, but we recognised that there was likely to be multiple long-term sequelae, and that we would need help, and we need help from specialists that really cover the whole spectrum of different possible outcomes from COVID-19 infection. So we had the patient really very much at the centre of the way we thought about this, and we were thinking about different types of presentation, and therefore what kind of systems in the body, organs in the body that might be affected. And that led to this working group structure where we then covered each of the different areas that we thought might be important. And we also linked in with the organ specialty charities to begin with, and then the long COVID charities later. And in addition to these working groups, we also had platforms to then be able to look at the data, the bioresource, and also thinking about imaging as well. The next slide. Our original aim, and you'll see from the later presentations how far we've got with each of these, were really some quite simple and overarching ideas. And these are things I'm sure you would be thinking of yourself. So our first aim was around what's actually the causes, so the mechanisms driving the consequences. So simple questions like, why do some people recover quickly, others don't? Why do some people develop other health problems later on? We wanted to understand about the impact of the interventions that were already being given acutely, and do they alter then the long-term sequelae? And could we also then think much more broadly by setting up a platform where we could then really have a much deeper dive into the science and understand what was going on? On to the next slide. So although there are our broad aims, very early on what we did with fantastic support from patients and also the James Linder Lines was able to then go through a priority setting exercise where we looked at a whole series of different questions and then prioritized them into the top 10. They're not in an order of 1 to 10, they're just the 10 that we felt had the highest priority. And they're shown here in their color codes really just to sort of fit into those groupings of looking at underlying mechanisms, diagnostic and prognostic tools and interventions. And this was published. It's you can access it through our website and also through the internet. And there's a whole series of other questions that were then raised, and all of the questions are planned to be answered, but these are really our main focus. So that's really the introduction. So I wanted to then turn to Rachel to then really start to put some colour to that and actually describe what have we found from the clinical data. So over to you, Rachel.
Thanks, Chris. And certainly we wouldn't have any clinical data if our patients hadn't participated. So we always want to thank the, thousands now of people that participated. Next slide, please. And next slide again. So this part is really to go through what did we actually do with the clinical data, and then Louise is going to take you through what we found with the bioresource and explain all the samples that we took from patients. So as Chris has highlighted, we had 3 clear aims. So our first aim was to look at what these long-term sequelae were and their mechanisms. Next slide, please. So PHOSP-COVID has now stopped recruiting, and our final cohort is nearly 8,000 participants, and they were recruited through hard work across 83 different sites with lots of different people involved. And again, many thanks both to the participants and the teams that did all the hard work on the ground. Now, to put our data into context, you do need to remember that our participants were discharged from hospital between February 2020 and March 2021. So they're largely people with the older variant. So this is all pre-Omicron and mostly pre-Delta. And also, for the majority of people, they contracted COVID before they were vaccinated. And then we had nearly 2,700 people that were recruited into what we called Deep Tier 2, which was where they underwent deep phenotyping. And these participants really provided a huge amount of time for us. These visits were getting on for 4 hours. Now, our cohort, again, to put the results into context, the mean age is 58 years old. So this is not an elderly population. They're working age, and indeed 2/3 of this population were working prior to coming into hospital. 40% were female, 75% white, 23% in the most deprived socioeconomic quintile. And overall, they had a high body mass index of 31. Now, this is the bit where I don't want to spend too much time being, the news was a bit gloomy, that actually not as many people were recovering as we'd hoped. So fewer than 1 in 3 participants felt fully recovered at 1 year. There was a median of 9 symptoms, and anyone here that has got lived experience with long COVID will really understand the burden of all of those symptoms. 1 in 10 were no longer working at the end of that year, and over 3 in 10 reported a change in work due to their health. There was minimal recovery between 5 months post-discharge and 1 year, and this is where we really need to crack on to get those interventions and treatments. There was considerable reduction in health-related quality of life, so whether how people feel in themselves at 1 year compared to their self-rated pre-hospital. 1 in 5 reported a new disability, and again highlighting this is a working age population. And the risk factors for non-recovery at 1 year were being female, being obese, and having been very unwell in hospital, receiving mechanical ventilation. But as whenever I'm speaking about long COVID, it's really important for us all to remember that anyone that contracts COVID can, sadly, is at risk of developing long COVID. Now, that cohort profile is actually now published in a preprint and is under review. Next slide, please. So we've done some further work, and when I ever say we, I really mean as the consortium, and this work was done by, or the large amount of work was done from Hamish Macaulay and Neil Greening at Leicester, and they actually looked at this concept of frailty, which is really a person's resilience to against other health outcomes, and most of the time frailty leads to poorer outcomes. We tend in the public to think about frailty as being in older people, but actually people with long-term health conditions can also have frailty. So we looked at it. This was one of the things that we had in our suite of measures from the beginning. And we looked at a particular definition of frailty and categorised people into robust, pre-frail and frail. And as you can see from the data in the left-hand side, less than 1 in 4 people were robust, like we would expect a working age group to be by 1 year. And in that pre-frail, there was almost 6 out of 10 people were in that pre-frail state, and over 1 in 10 were in a frail state. And those states were associated with reduced quality of life, which is reflected in that graph at the top, and they were least likely to recover in the pre-frail and frail group to the robust group. So this really then shows that there's a target for rehabilitation and recovery programmes, and further works going on around this, and also looking at the skeletal muscle in more detail, not just to describe the problem, but really to think about what those next steps of what we need to be testing. Next slide, please. There was work done by the team in Manchester, Callum Jackson and John Blakely, on sleep, and using the 24-hour physical activity monitors, which people wore for up to a couple of weeks, which also monitored sleep. And this team are really interested in the natural sleep patterns we all have, something called circadian rhythm. And they found in brief that in people with had been hospitalised from COVID, when they compared them to other hospitalised group or a non-hospitalised group, they were sleeping for longer. If you just go through, Tom, and just quickly add in the next boxes. Thank you. And the next one, thank you. But their sleep was less efficient and it was less regular. Next slide, please. And this was associated with symptoms of breathlessness, having reduced lung function, also anxiety and muscle weakness. And they did some complicated modelling around this. Now, what we really don't know is the impact of all of those things and how that circadian rhythm may also affect ongoing inflammation. And that team are going to look at the bioresource that Louise is going to talk to you more about and actually explore that more in a molecular level. The data does suggest that it's likely, we can't say that it definitely is, but it's likely that these problems on sleep are specifically or are a specific effect of COVID-19, and they may be amenable to interventions. And if we look at how we might improve sleep, we could also look again at those mechanisms. Next slide, please. In PHOSP-COVID, we utilised the imaging, the CT scans of the lungs that people had as part of post-discharge as part of their clinical care. And Ian Stewart and Gisley Jenkins led this work, really provided helpful information for people like me on the ground, trying to follow these people up and thinking how should we do that with imaging. And actually they found that just over 1 in 10 people that were discharged from hospital were at risk of having at least 1/10 of their lungs, that there was this abnormal lung pattern in there. And that's one of the first papers to really highlight how common that was. And again, there's further work from the fibrosis working group looking into the underlying mechanism of that in more detail, and again thinking about what treatments may or may not be needed. There's some good news today that the multi-organ MRI scans that was led by Betty Raymond in Oxford, and I hope you're getting from this the sense of how much of this work was led throughout the different centres at the UK, and she looked at how the multi-organ MR abnormalities compared with uninfected controls, and that's just been accepted in Lancet Respiratory Medicine today, we've heard. And the team in Nottingham have got a paper under review looking at a very detailed study, again undergoing multi-organ imaging with deep phenotyping of the skeletal muscle itself. They underwent an exercise protocol under MR, and they're reporting their results between patients and controls. Next slide, please. So leading to that second aim around trying to understand the impact of interventions both in the acute setting and at discharge. Next slide, please. We've looked at the effect of acute corticosteroids on the 1-year outcomes. The results aren't actually out in public yet, but they're due to be presented at the ERS this year, and we're busy finishing a manuscript at the moment. But the headline is, overall, there's either no effect or a very small effect, and certainly not clinically relevant. If you received acute steroids during your acute illness, whilst that is absolutely life-saving and really worthwhile, actually it doesn't seem to alter how somebody feels 1 year on. Next slide, please. And again, thinking about the health care pathways that people underwent in that early first year or two after their hospital admission, these were very varied over the UK, but that gave us the opportunity to map exactly what care people had at different sites. And this graph, at least, even if you haven't got time to synthesize all the different numbers, you can see it's quite different to some services offering no follow-up, no proactive follow-up, some offering complex assessment, complex intervention, and for both rehabilitation and mental health. And again, we're presenting the results of the clinical and cost effectiveness of those different pathways at the ERS in September. But overall, it highlights that if you adapt these services to the patient need, provide access to everybody, and then stratify on need, that those with the more complex multidimensional assessments and interventions do appear to be more clinically effective and look highly likely to be cost effective as well. And we've already fed that information up to NHS England. So thank you. And Tom, I think you're now going to play a video of Nikki describing how she's been involved in the PPI element of PHOSP-COVID.
I'm Nikki Smith. I had COVID early 2020 and still have various ongoing symptoms. Since having long COVID, I've worked with the NIHR and the World Health Organization, have co-designed studies and provided PPI, co-authored papers, and I take part in some studies too. I found out about PHOSP-COVID in 2020 when they invited the Long COVID Support Charity to be involved. I wanted to be part of it because, well, I'd never before been so ill that I really thought I was going to die until I got long COVID. And although fairly ill with acute COVID, it was the symptoms I was experiencing weeks and months down the line that were horrific, including having an oxygen level of 76% and waking up nightly gasping for air. Long COVID Support runs a peer support group with nearly 60,000 members. So we learn from both hospitalised and community cases about their symptoms, treatments, struggles, and their thoughts on research. And we hear from people who've lost their jobs, their homes, and worse. The importance of identifying the pathophysiology of this illness and potential treatments should not be underestimated. PHOSP-COVID brought together a whole raft of clinicians and researchers and injected into the mix those of us who are experts by experience. This resulted in a consortium with the knowledge and skills to research COVID-19 and its ongoing effects. I'm involved with a few studies and they all seem to be slightly different in the way they approach PPI. But the one thing that shows is that having the patient voice at the heart of research really helps ensure the condition is understood, the research remains relevant, and that effective results are achieved. I mean, I thought I knew what fatigue and breathlessness were like, but long COVID is a whole different level. And of course, those providing PPI on studies are not just patients. They may well have a lot of other skills too that can really benefit a study. One of the main tasks PHOSP-COVID asked us to assist with was identifying priority areas for research. So we designed and publicised a survey to get the views from a large number of people. And the findings were used as the basis for a study-wide event at which PPI members were involved as equals. That resulted in a list of questions that steered the subsequent research. But not only did PHOSP-COVID focus on the top 10 questions, all of the questions were made public so anyone could use them to advance research. The impact this illness has on people's lives and the economy is huge. So thank you to all of you who are conducting quality research with proper patient involvement. And please don't stop, because there are millions of people worldwide for whom this research must deliver.
Thank you. And I want to take the opportunity to thank Nikki and our members of the Patient and Public Involvement Group. I can see many of you on the call, and you've been really instrumental in keeping PHOSP moving forward and being relevant, for the patients that it serves. So thank you. We're going to move on to Louise's slides now. So Louise is talking about the bioresource and the samples from PHOSP.
Brilliant, thank you. So again, I'd just like to reiterate how incredibly grateful we are to our participants, and in this case, particularly those who not only agreed to provide their data and undertake additional tests and questionnaires, but also agreed to undergo some further inconvenience and provide us with biological samples, including blood, urine and oral samples. Next slide, please, Tom. So as you've already heard, participants attended 2 research visits, one at 5 months after they were discharged from hospital and one at 12 months. And at each of those visits, they were asked to provide samples, which were in addition to those that they provided for their routine blood tests. As many participants gave samples at both of their research visits, this means that not only can we perform sample measurements at a single time point after they were discharged from hospital, but it means that we can also look at changes between the 5 and 12-month visits. Furthermore, some participants had participated in other studies whilst in hospital, which meant that for some research questions, we are able to also compare their samples with those taken whilst they were very ill in hospital. Next slide, please, Tom. So there were 83 hospitals recruiting patients to PHOSP, and many of them, not all of them, were collecting samples from those that they were recruited. If we'd left the samples in storage at each of the hospitals that collected them, it would have been very inefficient for us to then run the sample analyses. So instead, what we did at intervals is we collected the samples together and shipped them together to a central laboratory in Liverpool, where they were logged into the central system and could then be easily accessed for analysis or research assays. And this also meant that we were able to keep a really close eye on the samples and do additional quality checks on them. Next slide, please, Tom. So these are the numbers of samples that were collected from our Tier 2 participants. And particularly for blood and urine, you can see that we've collected a very large number of samples, and that for a large proportion of participants, we have samples from 2 different time points. And we can extract lots of information from these samples. So for example, from the blood alone, we can collect DNA, information about how genes are functioning, and information about which proteins or molecules are circulating in the blood. And that can then tell us something about how that person's organs are functioning. Lots of people from the PHOSP team have been involved in the sample processing and analysis, and I will try to credit key people as I go along. Next slide, please, Tom. So as already mentioned by Chris and by Nikki in the video just now, in late 2020 through to early 2021, we consulted with patients, carers and clinicians to identify the most important questions that people with long COVID wanted us to answer. And there were 10 questions highlighted as being of highest priority. And those listed here are where the samples are particularly valuable when trying to answer them. You click on one, please, Tom. So in the next couple of slides, I'm going to show data from 2 studies that are attempting to answer these questions. Next slide, please, Tom. So whilst there's lots of similarities in symptoms between individuals with long COVID, we know there's also a lot of differences. So people might have different types of symptoms, for example, feeling breathless, fatigue, brain fog. Some people might have just 1 or 2 of these, whereas others have many more. Some people are more unwell than others, with the impact on their quality of life ranging from very subtle to hugely debilitating. And this means that we need to look at long COVID from different angles and perspectives to try and tease out why we see these similarities and differences in severity and symptoms. If you click one, Tom, please. So I'm going to show you 2 studies where we've tried to do this, specifically using the blood samples collected in PHOSP. Next slide, please, Tom. So inflammation. So very simply, inflammation is how your body's immune system responds to infection and injury. So for example, how your body responds to initial infection by the SARS-CoV virus and then to any lasting effects that the virus has had on your body. And in PHOSP-COVID, we measured 300 inflammatory proteins from the blood of around 900 participants 5 months after hospital discharge. Next slide, please, Tom. So using the physical test and questionnaire data collected in PHOSP, we were able to identify that our participants fell into 4 clusters which differed depending on how severe their ongoing symptoms were. So that's indicated on the vertical axis. And this was work that was particularly led by Rachel and the team in Leicester, together with Professor Ewan Harrison's team in Edinburgh. This cluster to the right in green was slightly different in that it also showed that there was a group of participants who had more severe cognitive impairment, or what we might call brain fog. And we wanted to know whether the difference in overall severity of symptoms might be explained by differences in ongoing inflammation. So to do this, click on one, please, Tom. We compared the levels of those 300 proteins in the individuals with the most severe impairment, so those in the red cluster at the top, with those with the mildest ongoing impairment, so those in this purple cluster on the bottom left. Next slide, please, Tom. And this is what we found. And these analyses were led by Leicester researchers, with particular thanks to our statistician, Dr. Matt Richardson. So this is what we call a volcano plot, and each dot on the plot represents one of the 300 proteins that we tested. So you can see a V shape being formed here, and the dots that are in red are the proteins that differ the most between those with severe versus those with mild impairment. And because they're to the right of the central part of the V shape, this means that they're found in higher levels in those with the most severe symptoms. And they're in red because they were so different that they met our strict criteria for statistical significance, which means that this is unlikely to be a chance finding. So the first thing to note is that there are many raised inflammatory proteins, which suggests that inflammation is related to why the people in this cluster still feel so unwell. Second thing to note is that amongst these specific raised proteins, there are proteins that were also found to be elevated in individuals with very severe COVID-19 in hospital. And there's previously been a drug approved that targets one of these proteins and was shown to be effective in reducing deaths due to COVID in hospital. So our data, supported by similar findings in other studies, suggests that similar drugs might also be of benefit to individuals with severe long COVID. And this is something we're working on now, and Rachel's going to tell you something more about this in the next section. Next slide, please, Tom. So I mentioned before that we can also look at symptoms by type rather than severity, and this might also give us important information. And this figure summarises results from a study that's so new it hasn't been published yet, but the findings have been made public. And this was led by Drs. Felicity Liu and Claudia Eustathiou and Professor Peter Openshaw at Imperial College London. And this study used the same 300 proteins and the same PHOSP-COVID participants as the previous study, but rather than comparing groups of people based on severity, they compared people based on the types of symptoms that they have. And this figure highlights the key findings for each type of symptom and lists the specific components of the inflammatory system that might be involved. So the letter and number codes as before are the specific proteins that were found at significantly different levels in individuals with these symptoms. And you'll note that some of them appear multiple times, but others are specific to different symptoms. Now, this is really early work and it requires further investigation, but it really highlights the message that not all long COVID is the same, and there is a need to understand this further in order to find the right treatments for the right people. Next slide, please, Tom. So the data I've shown you is from people who were admitted to hospital with COVID-19, but we know that most people were not admitted to hospital and that long COVID doesn't just affect those who were hospitalised. So what we're doing now is we're putting our samples together with samples from another long COVID study where most people were not hospitalised and with samples taken from individuals before the pandemic. And we're measuring these inflammatory proteins again at both 5 months and 12 months after COVID infection or hospitalisation. And this will allow us to better understand comparison, or better to undertake better comparisons between individuals with and without long COVID and people with different experiences during their initial infection. And importantly, this will help us to confirm that the findings from the previous studies are relevant to people who are not hospitalised and that the research we are doing is relevant to everyone. Next slide, please, Tom. So I've only had time to describe 2 of the studies, but these are some of the other ongoing pieces of research that I hope to be able to share more detail about at a future webinar. And answering these questions will take us closer to understanding the mechanisms driving long COVID and will in turn help us to find effective treatments. Next slide, please, Tom. So finally, I'd like to reiterate our thanks to the participants and research teams who took the extra time and inconvenience to provide the samples. These are extraordinarily valuable to research for long COVID. We have a large number of ongoing research projects that simply wouldn't be possible without them. And a reminder here that we have consent to follow up participants for 25 years, which means we'll be able to continue to make use of these samples and the data that they generate well into the future. So that's all from me. So I'm going to hand back to Rachel, who's going to tell you about the steps we're now taking to provide hope to people with long COVID. Thanks, Tom.
Thanks, Louise. And yes, this is really what we're all leading up to is trying to find that answer. And Louise highlighted then that it's got to be the right treatment for the right person. And that's the approach we're taking with PHOSP. Next slide, Tom, please. So that third aim was to try and do whatever we can to improve care for current and future patients. And that's where the 2 intervention trials that we've already got running from PHOSP-COVID fits in. Next slide, please. So we've got 2 intervention trials, the PHOSP-R or PHOSP rehabilitation trial that was funded through PHOSP and led by Enya Daines and the team through the PHOSP working group, some of here today. I'm delighted to say that they've done a phenomenal job and recruited around 160 people into this study. They've finished that, and I think the last person is in either this week or next week. And then they're looking at the results. And this intervention was really targeting when we had those 4 different clusters of severity, physical function was in there. So we very much targeted people with reduced physical function after a hospital admission to see what type of whether rehabilitation works, and if so, what type of rehabilitation. So the team has compared a face-to-face programme with a digital programme. And we're hoping that those results will be out in the autumn. As Louise also highlighted, in amongst those 13 red proteins was something called IL-6, and Louise highlighted that that's a molecule that's raised during the acute admission, and a drug called tocilizumab and others has helped in people with that ongoing inflammation in hospital. And we're looking at a study that we're leading from Leicester, but led with the involvement of the PHOSP-COVID platform, looking at a randomised control trial to see whether this drug in adults with long COVID and an inflammatory phenotype, so not in everybody, to see whether or not that will actually improve health-related quality of life after a 12-week course. It's literally in the setup phase. We literally just heard from one of the national governing bodies yesterday that they've looked through it all, and we've just got to respond to them, and we should be starting recruitment across 15 sites in the autumn. And again, we will be looking for patients, obviously, to volunteer to be part of that study. And we've talked about the importance of patient and public involvement, and certainly in that trial they have changed several things to do with the way that we're conducting that trial, and not least that we are now looking, you'll notice that it doesn't mention post-hospital, that we are looking at all people with long COVID and this inflammatory phenotype, and the team have very much encouraged us to do that from the start. I think that's back to you now, Lindsay.
Thank you. Can we have the next slide, please, Tom?
Oh, no, sorry. Apologies. So we had a PHOSP summit for a couple of days in end of March, and that's really, this webinar is to share publicly some of the findings that we're allowed to. Louise also covered that there's quite a few different analyses that have finished but haven't yet made it to the public domain, but are certainly on their way. So our summary so far is that we've been able to carefully describe the post-COVID sequelae up to 1 year post-discharge, and PHOSP does remain the largest detailed cohort internationally. We've got novel data on this measure of frailty, breathlessness, which I haven't shown you today, and imaging abnormalities, and those already are being fed back into the policymakers to help clinicians like myself improve clinical care if we know what we're actually looking to find in clinical care to target. Louise has highlighted those first steps to understanding the underlying biological mechanisms to lead to potential treatments, that we've shown that we've got these inflammatory proteins associated with disease severity, and also intriguingly that they also may be relevant to particular symptom groups. Our data have highlighted potential modifiable targets that we're cracking on and then testing in clinical trials, such as systemic inflammation and reduced physical function. And that data will soon be out around the healthcare pathways, which healthcare pathways were clinically effective, but also good, meaning which clinical pathways help people feel better, but were also value for money. And all of this data we feed back really iteratively. We don't wait until we've got the final version. We discuss with the NHSE Long COVID Task Force, we input into the Long COVID service specification, and we've also fed back to NICE guidance. So that, like I say, that's really been a very iterative, fluid process of dissemination. And now I am handing back to you, Lindsay.
Thank you. Next slide, please. Yes, we've come to the panel discussion. So if you have any questions or comments for the panel, then please post these in the chat. By all means, tell us about your experiences, but we can't address questions about your individual clinical care on a public webinar, I'm afraid. So if we just go to the next slide, please, Tom. We did have some questions that were submitted through the registration process, so it's probably helpful if we start with these whilst you're putting your thinking caps on. So Chris, could I come to you first of all to answer the question about new studies that people can get involved with and what the plans are for PHOSP-COVID in the future?
Yeah, incredibly important that studies are coming through and there's real opportunities. There's different approaches to the studies that are currently ongoing. Some studies are then looking at repurposing existing drugs, such as things like antihistamines or common anti-inflammatory drugs, and then giving that to large numbers of people without targeting the individuals. But that kind of platform approach may then start to reveal where there's benefit, and it can then help to then try and identify in whom. PHOSP-COVID has taken a slightly different approach. So we're, as Rachel's described, very much looking at individual problems that we've identified through those mechanisms that Louise has described, and that then leads on to a more targeted approach. So I think that will help with certain individuals that then have had long and still have ongoing long COVID, with particularly those with inflammation. So they're the current approaches. The rehabilitation you also heard about. There are some interesting studies coming through around trying to alter the metabolism of the body with drugs that so far have been used for weight loss or diabetes, some interest there. And there's some new drugs as well around trying to alter what we call oxidative stress, so the way the cells control energy within the body, and that's also being used to try and target fatigue. So there are a number of different opportunities, not just within PHOSP-COVID, but looking much more broadly. And the UK is very much taking a lead on this, and there's quite a lot of things coming through, and I'm optimistic that will translate into treatments. And I suspect probably not treatments that will treat all of long COVID and everybody who's suffering, but start to help with particular problems in particular individuals towards that what we think of as precision medicine approach. And I think that's really where there's going to be the greatest gain in trying to tackle long COVID.
Thanks, Chris. And the next question is about patient and public involvement. So I think I could answer that one. We absolutely are going to continue to include patients and members of the public in the work of PHOSP. That's already ongoing with the new intervention studies that you heard of from Rachel. So patients have been involved in developing the protocol and will be involved going forwards, looking at things like patient information sheets, making sure that they are in lay language and that we're not putting too much burden on patients with research visits. Patients are also involved in any dissemination that we do, so any newsletters, any publications. We've got a mechanism now to include patients as early as possible in looking at data. And, you know, I only see that going from strength to strength as we continue with PHOSP. Sorry, go on, Rachel.
Oh, I was just going to say, maybe you were coming to that, but to invite Claire to talk about long COVID support, where that comments around where they can meet up.
Yeah. So Claire, do you want to talk about long COVID support and any other charities perhaps that people might want to tap into?
Yeah, so speaking for long COVID support, we have an online support group. I'm afraid it is based on Facebook. So if you don't have access to that, I do apologise. But within that, so that runs 24/7, 365. Every single member is screened before they can join. It's a private group. It's a safe space. Every single post is monitored by our international team of moderators to keep information. You know, we kind of keep it a safe space in terms of information. We don't allow quackery or promotion or anything like that either. So there's an ongoing dialogue in there. We also host lots of online events which people can, you know, drop in chats, all held online so people can join from their beds, as well as an active programme of social and wellbeing events, including our long COVID choir, chair yoga, all that sort of stuff. There's an equivalent organisation focused on children, long COVID kids, who do great work, and I'm sure there are other support groups out there, some of which might be face-to-face in local areas, but I'm afraid I can't speak for them. But if you want information from our organisation, head to longcovid.org, which hopefully you can see the link on my backdrop as well.
Thanks, Claire. I'm going to come to a question in the chat now. It's for you, Louise, about obviously the hospitalised samples came from spring 2020 to 2021. Do the 500 community cases come from the same time period, or might they represent different variants?
Yeah, that is a really good question, and it is something that we were having to really think about when we were designing the study and the collaboration. So there is some overlap that we've tried to maximise when selecting individuals. The other study is a very large study, lots and lots of individuals. It also differs in terms of the people that were in it, so they're a slightly different population to our population, so we're taking that into account when we're running the analyses. But both studies will allow us to look at changes over time as well, so we'll be able to factor in differences of the period that the variants that people were exposed to. There'll also changes in vaccination as well. So the advantage of doing a really large study is that we can to some extent try and understand the impact of those different variables that we couldn't control for at the time. I hope that answers your question, Jenny. Thanks, that's a really good one.
Yeah, thanks, Louise, and yeah, great question. Rachel, can we come to long COVID assessment now? So first of all, I guess, is long COVID acknowledged as a separately identified condition now? Would be the first one.
Absolutely. It's defined, as I expect people know, both predominantly from the patient and public definition of long COVID, symptoms lasting 4 weeks since contracting the virus, and then the WHO definition and then the NICE definition. So although it's important for clinicians to exclude other conditions that might be driving those symptoms, long COVID is a diagnosis and has relevant codes that should be used. The questions around whether people should have a comprehensive neurocognitive assessment and actually what's being done to assess that. From a PHOSP perspective, we've got an affiliated grant through the Brain Working Group and the Wolfson Foundation and MQ doing far more detailed testing in hundreds of our participants. That data has been collected. Again, it will be a very large data set, and there's some imaging alongside that, and a team are analysing that at the time. In clinical care, there are assessments around neurocognitive that we do, and actually the NIHR locomotion study are looking at what tests for cognition are really the best to use in clinical care, so they're doing that. The questions around whether conditions that you've had before can worsen after COVID, yeah, absolutely. It's quite common that pre-existing symptoms also worsen alongside new symptoms, and the comment around diabetes, again through PHOSP, the diabetic or the metabolic working group are looking through data, and there's other data coming out, and again with the cognitive work, I should also flag that there's other studies in the UK that are really starting to get their results out. So I'm really hopeful over the last few months we should hear much more about the neurocognitive assessment and how long those symptoms last, to answer the last question of the long COVID symptom portion there.
Thanks, Rachel. Well done. I'm not sure who wants to take the treatments section, but there is a question in the chat from Deborah as well about wanting to hear more about suggested treatments, and via the long COVID clinic, she's had an offer of counselling which has had zero impact. So I don't know if anyone wants to comment on what treatments might be the most effective.
I think that's what Claire's got her hand up. Yeah,
we're all going to jump in, aren't we?
Yeah.
Clearly, I'm not a clinician, and I obviously bow down to other people for that. But just to say that in long COVID, there are certain conditions which are very, very commonly diagnosed as a result of long COVID, or whether they coincide or cause versus coincidence, whatever. But one of those is dysautonomia, with POTS a particularly common subset. There was some research published recently of whom almost 80% of the sample of the cohort in the study had signs of POTS. And that is something that can affect your heart rate, your blood pressure, people feeling dizzy, passing out, all sorts of things. And actually, there are things that can be done to help with that, both pharmacological, but also interventions such as consuming electrolytes and wearing compression garments. So that's one example. And there's actually a question on the slide, isn't there, at the top of the question, asking about research into histamines. Histamine intolerance is also incredibly common among people with long COVID, people developing intolerances to foods, their pets, cleaning products, household products, all sorts of things, including the sun. And so taking pharmaceutical interventions can help with those, such as H1 and H2 blockers, and they can also have the benefit of dampening down your wider long COVID symptoms. They don't work for everybody. There are lots of different types of those, but those are two very common areas where if someone has long COVID, they might want to raise with their medical practitioners. Could they be experiencing dysautonomia and POTS? And could they be experiencing histamine issues?
Chris, do you want to add
I
anything?
think that was a great answer, and on a similar theme, I agree. It's about unpicking what's actually the problem for an individual. So as Rachel showed, there's that data that's come out recently around sleep disturbance, and it clearly shows that seems to be quite a specific problem that happens in people with long COVID, and there's NHS supported app on Sleepio, which is then really trying to help with improving your sleep quality. So that could be another strategy. You heard about the rehabilitation, and clearly exercise in some individuals can be the wrong thing to promote because they can get post-exertional malaise. But we identify that it's likely that many people do benefit. So it's then trying to understand within an individual what current drug treatments, what interventions from more of a psychological perspective or from an exercise and lifestyle perspective may actually be very helpful, at the same time as us trying to work faster and smarter at getting these new mechanisms identified and these new treatments developed. And I agree with we feel equally frustrated that these things never happen as fast as we wish.
Thank you. Anything to add, Rachel?
No.
No, super. Okay, I'm conscious of the time. If we didn't get to your question, I do apologise. Can we just go to the last slide, please, Tom? So we will be sharing the recording with you by email, and it'll also be available on the PHOSP.org website. And we'll also be asking for your feedback to help us in planning future events. So if you do have any questions that we didn't get to cover today, then by all means do send them by email, and we'll get back to you. So thank you very much for joining, and see you all for another webinar soon, hopefully. Thanks.
Thanks, everyone.
Thank you.
Thanks, everyone.
Thank you.
Thank you. Bye.
Thank you.